Hi, welcome back to Chapter 10. I want to finish up the sensory organs, the sense of hearing, and sense of taste, and the sense of smell. So let's talk about these structures first for the ear and talk about how sound is perceived. The ear is not just for hearing, there's also structures that give us our sense of balance. Let's go ahead and look at those now. There are three parts of the human ear, and let's work from the outside inwards. The outer ear is involved with hearing, and it's composed of the auricle or pinna, and that's the part on the outside that you might get pierced. The external auditory canal, this is where water gets stuck when you swim, and it ends at the tympanic membrane or eardrum. The eardrum is made up of a thin layer of connective tissue or CT that vibrates in response to sound waves entering the canal. The eardrum transfers sound energy to the middle ear, which contains a set of bones called ossicles. The eardrum marks the boundary between the outer ear and the middle ear. The middle ear is involved with hearing and amplification of sound. It's composed of small air-filled mucosal-lined cavity that leads to the oval window, and that leads to the inner ear, which functions in both hearing and equilibrium and is made up of mostly neural structures. Here's a closer look at the ossicles found in the middle ear. There are three bones called ossicles in here. The hammer, or malleus, is the first bone and is physically connected to the tympanic membrane. The hammer moves and it hits the anvil, or incus, and that connects to the stirrup, or stapes. Latin terms are often interchangeable here. The stapes, which is physically connected to the oval window, connects to the middle ear. It's set up deep inside the skull. You can see the bones surrounding this inner ear and middle ear portion. And that leads to the ear's neural structures, which are the actual hearing part of the ear. This is also where our sense of balance happens. In the inner ear, it contains sensory organs for hearing and balance. The receptors located in the inner ear for hearing and balance respond to separate stimuli and are activated independently, and we'll look at these here in just a minute. First, let's look at the inner ear. This is the cochlea. This is the hearing part of the ear, and it looks like a snail shell. There are semicircular canals out where the antennae of a snail might be. The utricle and the saccule are the balance part of the inner ear. The cochlea, which is the snail shell part of this organ, is fluid-filled and takes on a labyrinth of a tube within a tube series of structures. The inner tube contains endolymph, and perilymph surrounds that. The organ of Corti serves as the sensory organ. This receptor inside the cochlea has hair cells that get triggered by the movement of the fluid. The hair-like structures serve as nerve receptors for both hearing and balance. To transduce sound, the mechanical energy from the movement of the middle ear ossicles pushes on the oval window leading to the cochlea. This force moves the cochlea's fluids that, in turn, stimulate the tiny hair cells. The oval window, which is in direct contact with the stapes, attaches to the scala vestibuli, located at the base of the cochlea. Vibrations at the oval window induce pressure waves in the perilymph fluid contained in the scala vestibuli. The scala vestibuli and tympani are continuous all the way to the apex or point of the cochlea. Sound waves in the vestibuli pass through the scala tympani, which displaces the round window located at the base of the cochlea. Here's a diagram of sound waves moving through air. Remember we did this in lab with the Weber's test. You had sound frequencies that you were listening to. So low frequencies, which is a measure of pitch, can travel all the way through the scala vestibuli and into the scala tympani. As sound frequencies increase, they are transmitted directly through the vestibular and basilar membranes toward the scala tympani. As sound waves move and enter the ear canal, they become amplified once they hit those bones of the middle ear. See how the peak height increases here? This amplified peak height continues in the middle ear's fluid-filled tubes. If we straighten out that snail shell-shaped cochlea, we can follow the sound waves through this tiny pea-sized fluid-filled structure in the inner ear. High frequencies produce maximum stimulation on the organ of Corti, which is located closer to the base of the cochlea, and lower frequencies stimulate closer to the apex. We can read this figure by following the color-coded frequencies through the flattened-out cochlea. Frequency is measured in hertz, abbreviated Hz. The highest frequency is 20,000 hertz and only travels in the base area. But if we follow the lowest frequency, shown here in blue at 500 hertz, these low and slow sound waves permeate the entire length of the unwound cochlea toward the apical tip. Now here we can take a look at the hair cells. We mentioned these earlier in our description of the working parts of the inner ear. They will bend toward or away from the taller stereocilia on the right and side of this receptor. Depending on the pitch and frequency of the sound different, hair cells will move, but bending the stereocilia, there starts an increase in the amount of neurotransmitter release in the inner ear cells, resulting in greater generator potentials produced in the sensory neuron. Cranial nerve 7 is associated with the sensation of sound and balance. On the far right over here on this figure, notice that the bending of the smaller hairs are moving away from the taller stereocilia, and that shuts down the firing frequency of the action potentials being sent along the nerve. Less neurotransmitter is being released, and the frequency of the action potentials becomes slower. When the shorter hair cells bend towards the stereocilia, the calcium channels open and rapidly depolarize the receptor to fire more frequently along the nerve. Here in figure 1048, this is a great picture of the cochlea. And the utricle and saccule structures, circled here in red, are used in the sensation of balance. The saccule and utricle house the equilibrium receptors called the maculae, which function by responding to gravity and changes in the position of the head. Moving on to figure 1049. These hair-like structures are part of the vestibular apparatus, where the utricle and saccule provide information about linear acceleration. Remember the semicircular canals that make up the snail's antennae? They're oriented in 3 planes, X, Y, and Z, in order to give the sense of angular acceleration in the head. Who actually did the spinning stool test for nystagmus in the lab? When these stereocilia are bent in the direction of towards the tall kinocilium, shown here in pink. Think of kinetics when you hear this term, because we're talking about movement now, even though both examples were using hair cells. When the hair cells move toward the tall kinocilium, depolarization occurs and neurotransmitters are released, which stimulates cranial nerve 8, which is the vestibular cochlear nerve. When the hairs are bent away from the kinocilium, the receptor becomes hyperpolarized and becomes inhibited. In this way, the frequency of action potentials in hair cells carries information about movement. Now let's talk about gustation and the sensation of taste. This gourd-shaped receptor buried in the surface of your tongue is responsible for the sensation of taste, called gustation. It's functionally based on chemoreceptors that respond to a variety of dissolved chemicals in aqueous solution, aka your saliva. There are about 10,000 taste buds on your tongue. And there are 5 basic taste sensations: sweet, caused by sugars, saccharin, alcohol, and certain amino acids can give the sensation of sweet. We can perceive salty. These are usually metal ions like sodium. Sour, which is a hydrogen ion like found in lemons, and that's usually a low pH, so high hydrogen ion concentration causes the pH to be acidic, and we perceive that as sour. Bitter would be caused by alkaloids, such as quinine and nicotine. Those are going to be high pH. And then the sensation of umami, which is savory, brothy type flavors, usually caused by amino acids, and we get these sensations from meat. Binding of the food particles depolarizes the taste cell membrane and releases neurotransmitters and initiates a generator potential that then elicits an action potential along cranial nerve 9, shown here at the bottom. Okay, so the sensation of salty and sour flavors, both of those are next to each other here on the left. These flavors operate by activating ion channels that depolarize the receptor. Salty flavors from sodium and sour sensations from acidic foods like lemons that have a hydrogen ion both cause a depolarization through ion channels. Sweet, bitter, and savory flavors all activate G protein linked receptors and use secondary messengers inside the cell to stimulate the release of neurotransmitters. These three flavors are together on the right side. You can draw a line between these four examples. The two on the left for salty sour use the same cellular pathway, and sweet, umami, and bitter here on the right use the G protein pathway. This might make your studying a little bit easier. The hypothalamus and limbic system are implicated in the appreciation of taste, but we can't ignore the fact that temperature and smell are also a huge factor that goes into the way we perceive taste. Now let's look at the perception of smell through the process of olfaction. The organ of smell within the nose is all of the olfactory epithelial, so the skin, the mucous membranes, the epithelial lining of your nose. The olfactory receptor cells are bipolar neurons that have cilia attached in order to catch the odorant particles floating around in the air. The olfactory receptors are surrounded by supporting cells that cushion them, and these are the basal and sustentacular cells, circled over here in red on the right. Notice how the axons of the olfactory nerve, cranial nerve 1, have to extend up through the bones and the sinuses in order to carry the signals to the olfactory cortex and the limbic system. Here is a transmission electron micrograph of an olfactory neuron. Basal cells act as stem cells that produce new receptors every couple of months. These are the only neurons that have the ability to regenerate on a regular basis. All other neuron types are incapable of regeneration due to the lack of growth hormone receptors and because of their highly specialized nature. The supporting cells that contain detoxifying enzymes, these also help keep the membranes naturally cleaned out. Olfactory receptors respond to several different types of odor-causing chemicals, but the physiology of signal transduction causes a G protein receptor and cyclic AMP as a secondary messenger in order to open sodium and calcium channels, causing depolarization of the receptor's membrane, which in turn triggers an action potential. Let's take a look at that process. Here we can see the signal transduction process that involves the activation of adenylate cyclase at the membrane, which triggers the activation of cyclic AMP. Notice how the operation of these ion channels allows both sodium and calcium ions to flood inside the cell, causing massive depolarization along unmyelinated axons that terminate in the olfactory bulb.